and have held executive as well as technical operating roles positions in the companies like Microtherapy Research Labs, Chennai, Vanta Biotrans, Kemin Industries, Chennai, Biocon Bristol, Mysore Skib Research Center, Bangalore, Origin Discovery Technologies Limited, Bangalore, and JRDS Research Center, Ahmedabad. Dr. Deepak Barot has extensive drug discovery research experience in the field of metabolic disorders, inflammation, oncology, and neuroscience. He was the DMPK scientist involved in generating early ADME and IND enabling studies data for saroglitazar, the first drug from an Indian pharmaceutical company, which was approved by DGCI in 2013 for the treatment of diabetic dyslipidemia and hyperglycemia. Dr. Barot was the key person involved in the development of Cariolect, one of its kind water-soluble enzyme mixture for improvement of digestion for veterinary use, and his laboratory did all the required in vitro and in vivo studies and field trials of Cariolect that led to its successful launch in 2016. With his overseas collaborations and extensive industry network, he is involved in bringing novel human-based in vitro experimental model, especially primary cultured human hepatocytes and most recently enterocytes, to Indian CROs and research institutes. He has been associated with all areas of drug metabolism, pharmacokinetic, pharmacology, and toxicology, lead optimization, preclinical pharmacokinetic, pharmacology, toxicology studies, and pertaining to the drug discovery of new chemical entities. He has broad scientific knowledge, leadership skill, and hands-on experience in conducting DMPK, pharmacology, toxicology studies, and various analytical methodology used in laboratory settings such as PUPLC, LCMS, etc. He received his PhD from Anand, and most recently he has been appointed as a cluster head, Central and Western Region for License Sector Skill Development Council, New Delhi. And he was instrumental in initiating and bringing the APA meetings to India, and is currently a scientific advisor to Boston International Conferences, Boston Society, USA. By the way, he obtained his BVSc and AH, MVSc, PhD from Anand. Myself and Dr. Bhavsar has taught him at all the levels. He was General Secretary at the time of undergraduate studies and dynamic and was anchor in Amul Volcano Cultural Festival of all the colleges around the Anand. He is a good orator, speaker, and in short, all-rounder. He is a successful educator. I will remind to those who have attended the ISPT 2018 in the Anand pre-conference workshop on 4th December was held at his institute, that is the Prerna Bhav Innovations Private Research Laboratory. And so that way, it's a worth to listen him about the drug metabolism and pharmacokinetics. One more thing I would like to say that ISPT 2020 announcement is already there, and I hope all of you are aware of that one, and we will start registering for the that e-conference. And to the younger scientists, please participate in the e-poster competition which is being held, and the last date for submission of the PDF form of your e-poster is the 20th September. Kindly do needful and register in maximum number. Once again, I thank the Srinivasan for giving me the opportunity to introduce my student, our student. And in genetics, we say that the next generation is about the parents, and that way I am happy that our student, Dr. Barot, has become a successful entrepreneur, and he is going to deliver his talk today. I hand over mic to Dr. Barot, please.
Thanks so much, sir. Dr. Deepak Barot, you may share your slide. I'll just make you.
Hello?
Yeah, Dr. Deepak. Yes.
Is my screen hello?
Hello. Yeah, Dr. Deepak, your slide is visible. Please put it on the slideshow.
Okay.
I request all the members to please mute your audio as well as don't stream your video because that will consume a lot of bandwidth. So that will disturb the presenter while presenting it. So kindly mute your audio and don't stream your video, please.
Okay. So first of all, would like to thank you Dr. Thakur sir for such a kind words and an introduction. Thanks a lot. Actually, it really took me back to my college days, and it's an absolute honor for me to get this opportunity to present at a forum of ISVPT. So thank you, Dr. Thakur sir, and would like to take this opportunity to thank all the organizing committee members, especially Dr. Srinivas, for inviting me to speak at this prestigious platform of ISVPT webinar series. So thank you once again. As I said, it's an honor for me, and in fact, on a lighter note, I'm as nervous as almost 25 years ago when I was appearing for my Viva examination. So it is same nervousness I'm feeling today. So thank you, and I'm really looking forward to this, and would like to thank all the organizing committee members for this opportunity. So Dr. Srinivas, if you can just confirm if slides is visible.
The slides is not in slideshow format. It is in still.
Is it?
Yeah. Could you make it a slideshow? F5. If you press F5, it will work on. I don't know. Maybe you can. In Cisco Web.
Deepak, make it full screen.
Yes, exactly. Dr. Srinivas, can you just quickly remove the presenter, this thing, just for a second? I think I'll be able to, because of the, okay, I should be able to do it now, I guess.
Just I removed it. I'm doing it again.
Okay.
Yeah. So let's try now.
Okay. So is it visible now?
Not yet.
Okay. 1 second. Yeah. I hope now is it a slide mode?
Yes, it is in our screenshot. Yeah.
Okay. So sorry about the slight delay in this. So first of all, thank you all organizers, and as already I think Dr. Thakur has described about the DMPK. I think drug metabolism pharmacokinetics is the basis of drug discovery and development. And since last 20 plus years, it has been a dream run for me into the field of drug metabolism and pharmacokinetics. In fact, when I was pursuing my master's, I used to dream that, okay, one day I'll get into DMPK division and I'll get to do some research into it. So I'm thankful to all my mentors and especially my institute for actually helping me gain that required qualification and knowledge, and so that I was able to get into DMPK division many years ago. So already Dr. Thakur has mentioned about my industrial experience so far. So since I was told by Dr. Srinivas that the audience is majorly masters, MBSc or MPharm students and PhD scholars, so I just thought that let me just put up the slides together in a way that we can have a brief overview of DMPK in drug discovery and development scenario. So basically, we study all these things in theory, but I would like to focus more on, from an industry perspective, when you're running drug discovery and development projects on any disease targets, So these are the common assays which we generally run. So I thought since mostly researcher students would be there, so it would be helpful for them to really understand the application part of DMPK into drug discovery. Okay. So with that, let me start. So I'm going to take you through this presentation about the process, drug discovery development process as such. What are the in vitro ADME assays? Key pharmacokinetic parameters. We all know, I mean, most of the students might have done their research in PK, but from a drug discovery and development point of view, when you're working on a project, when you're working on drug development, what are the important PK parameters and how do they correlate? Then in vitro tools into drug metabolism, which are routinely used in early drug discovery. And then we'll talk about discovery bioanalysis as well, because this DMPK assays or even efficacy can be completed unless there's an analytical portion attached to it. And then finally, very quickly, in just a couple of slides at the end, I would like to talk about council, life science sector council, so that you will have a fair understanding about this initiative of Skill India from Government of India as well. So let us start with this small cartoon, like innocent kid. When doctor is ready to give a shot, he's just talking to his mother, saying that how will that stuff get down there up to my sore throat? So from the injection site, how is it going to reach my sore throat? So that's pretty much the DMPK uncertainties and lack of understanding actually. So very quickly, this is the PBR. Presently, my association is with Prerna Bioinnovation Research Center. This is situated in Gandhinagar, in the capital city, in the state of Gujarat. Again, it's a very small CRO and completely into drug discovery and development contract research. It has been started almost 6 years ago, and the aim is to provide end-to-end solutions in preclinical scenario. And we also work into clinical development as well. So this is just a few glimpses of the lab, how it looks. We do have CPCSA approved animal facility in place. And of course, it's a conventional animal facility, but sufficient enough to actually do all kind of early drug discovery projects. So of course, we have been working on quite a few many interesting projects, but very quickly, since it's an ISUPT forum, I just thought I'll share one of the success stories we have worked on. So this is an enzyme mixture called Cariolact, which was launched almost 2 to 3 years ago, and it was one of its kind water-soluble enzymes for improvement of digestion in milking animals. So basically, it was improving the efficiency of milking animals, and it was a cocktail of all the enzymes as mentioned. So the product was launched on World Veterinary Day in 2016. So we developed this product in collaboration with a biotech company from Hyderabad, and quite a few many studies were done to prove the efficacy and the safety. And finally, it was launched on the World Veterinary Day. So, of course, it was newly covered in media as well, because this was purely, the aim was by improving the digestion efficiency in milking animals, the overall outcome was improvement in efficiency of the animal, increase in fat percentage, increase in milk production, yield. So that's why it was duly covered in the media as well. And I'll just cut down to the most recent one. Just a couple of days back, we have launched a very portable small unit of UV disinfection box. This is keeping in view the current challenges with pandemic crisis situation of COVID, where we just need to be careful. We need to be safe. So it's a portable UV disinfection box, which can be, and it's quite affordable, and every non-living thing can be sterilized in it, so you don't have to use any chemicals or disinfectants. In fact, it just just do this job by UVC. And again, it's not a new concept. We have been using UV laminar flows for many, many years now. So I think this was just a portable version of that, which we launched just a few days ago. So with that, I think let's jump into the topic for today. So I would like to start with drug discovery and development. So from my experience of whatever couple of decades of experience, I think it's been a roller coaster ride. Be it from the starting point when you actually go out and look for a clinical need, first you need to establish a clinical need before you really start working on any of the projects. From there, when you work on whether there are any opportunities to do the patenting, and then you plan your preclinical studies, starting from proof of concept to ADME, optimization to efficacy to toxicology, safety studies, keeping all this thing in the regulatory framework you have to keep in mind. And eventually it has to have a business strategy in place. Otherwise, even if a good product can fail, of course, a lot of finance gets into it, a lot of chemistry efforts in product designing. Eventually, after preclinical phase, we take it into clinical trials. And then once it is launched into the market, that's the time you look for return on investment in form of reimbursement. So basically, it's a roller coaster ride because your drug can fail at any of the stages. There are challenges at every single point in this. So that really makes this really interesting field of drug discovery and development. Of course, there used to be a time when it used to just happen by chance, discovery. But nowadays, with our advancement and more understanding of ADME or efficacy, pharmacology, safety mechanisms, now I think it can be done in a more structured manner. So again, just a cartoon which says, a patient is saying, I stopped taking medicine because I prefer the original disease to the side effects. So this is the crunch of it, because with so much of advancement in the field of drug discovery and development, even today, the major issues are revolving around side effects, right? So that's what really makes this field really interesting. So when you talk of drug discovery and development, of course, all of you are aware of discovery, preclinical, clinical, all the way to the market launch. But just I wanted to emphasize here on the development process is almost 10 to 15 years. Okay, so it's quite a long process and huge investment. We're talking of $2 billion of investment. And what are the chances of success? It's just 0.003%. In subsequent slide, I'll show you how does it arrive at 0.003. That is 1 in 30,000 molecules will actually make it into market as a drug. So regulatory agencies, pharmaceutical companies, everybody really wants to emphasize on two aspects. We want to have a safe and efficacious drug. So actually, how do we really develop better drugs with more success? That's what this whole research, drug discovery development efforts is all about. Okay, and would like to emphasize that there was a time when drug used to fail because of poor ADME properties. I would rather say because of our lack of understanding of ADME principles. So there was a time back in 2001 when more than 40% of the drugs used to fail because of poor ADME properties. But by 2007, almost a decade ago, this number has come down to almost 10%. Okay, and by 2011, it is 7%. So it proves that our overall understanding of ADME has improved a lot. So we have come a long way in our understanding of ADME principles. Okay, so this is the funnel, the most famous funnel for the drug discovery. You start with thousands of molecules, end up in just one approved drug over a period of 10 to 15 years. It's a costly affair, of course. As you can see, the investment in terms of R&D expenditure all the way from 1995 to 2014, you can see it has always increased. Okay, so it is of course a costly affair. How much it costs? So if you see for a preclinical phase alone, around $335 million we are talking about. Clinical trials, even more than that. And even post-approval, you need $100 million alone. So that's why this whole process is very costly. And I was just talking about the probability, like why only 0.003%, because this is just 30,000 compounds you synthesize. You come up with just 2,000 will enter into preclinical. Before that, during your in silico itself, you will drop down that many molecules. From that, hardly 200 will enter into phase 1. This is also quite optimistic. Phase 2, phase 3, finally, once you see the compound which will make a return on investment would be hardly one molecule. So that's how this is 0.003% of success rate for a successful molecule into the market. So attrition, when you say the late clinical phase, is very expensive. So we should always remember that it's not about our investment on the drug which is successfully launched, but also the investment into the drugs which never made it into the market. So while initial discovery phase itself, if we don't pick up these drugs early, then even those drugs are going to cost you when they fail at a later stage. Yeah. So as we already seen the investment, like it has always increased. Every year, the investment on R&D has been increasing. We already seen the reasons for the failure, as from 40% of poor PK used to be the reason for failure. Now we have come down to almost to 7%. But even now, the majority of the drugs which fail is because of toxicology reasons. Yeah, even the clinical safety or even efficacy. So many times it is the efficacy of the drug because of whom the drug fails in the clinical trial. Yeah. So when now you look at it, I hope you will understand this funnel more comfortably. That when you start with 100 discovery approaches, different programs, finally you end up having only one product, and by then your entire drug would have taken the journey all the way from screening to preclinical pharmacology, to safety studies, clinical studies, and the safety study in the clinical scenario, and finally completed molecule which actually is launched into the market. So that's how this takes this long period. Because we hear all this time that why does it take so long? But this is the reason why it is taking so long for a drug to get into the market. And I don't want to really discourage the young scientists, but I just want to showcase this as well, the other side of it also, that after having so much of research, so much of our understanding on drug discovery and development, when finally a molecule is launched into the market, this is also a scenario which we should always keep in mind. These are all the drugs which are withdrawn from the market. So even after doing this many studies, so much of investment, a drug is being launched, and then also it has to be withdrawn. And as you can see, there are various reasons, starting from cutaneous reaction. Majority of them you will see because of hepatotoxicities. Many of them will have risk to the heart diseases, QT prolongations. So these are all the reasons, because in spite of screening of molecules for all these parameters very early in drug discovery and the development phase, still there are chances that when a drug is launched into the market, eventually we may end up after a year or so, and then we understand that there are some life-threatening side effects, and for that also we have to withdraw a drug. Okay, so again, of course, I wanted to talk something about veterinary drugs as well, but it's not in the scope for today's presentation. But just wanted to, in a couple of slides, wanted to showcase that when you are developing a veterinary drug, because all the scenario which I mentioned was about a human drug, but for a veterinary drug, these are the major 4 critical standards from a safety perspective. Is it going to be a human food, the target animal, environmental, and user safety? So these are all the criteria, including effectiveness. Then we want to have a quality manufactured product and label, because that also forms a very important aspect of a veterinary drug. So these are all the criteria or the standards where you can actually develop a drug for a veterinary use. So if you see, already we have seen this. This is the whole scenario. for a human drug. Now, exactly same is the development process for a veterinary drug. In the next slide, if you see here, so more or less it is just the same with a few differences. As you can see, of course, discovery and the proof of concept is important aspect even for veterinary drug development process. There is a whole development thing. Regulatory submissions happen, and like we have NDA submissions for human drugs, here it is called NADA submissions or approval to CVM or FDA when you apply for approval of your drug. So all I wanted to emphasize is more or less the process is same. Okay. Of course, particular assays requirement for human would be slightly different than for veterinary application, but overall drug discovery development process in general are just the same. Okay. So I've taken this slide from the FDA, and they have initiated their new effort, which is basically to support new idea generations for veterinary medicines. So this is where CVM, they also interact at different phases of drug discovery and development process for development of veterinary drugs. So let's talk about drug discovery and development I'll just spend a couple of minutes on this slide because this really defines the entire process of drug discovery and development. Okay, so if you see, you want to have a targeted drug, okay, because if there are any off-target effects, you would like to avoid that. It should be selective, again, related to your selectivity to your target. It should be highly potent. It should be physically stable so that manufacturing becomes easier going forward. It should be soluble, permeable, metabolically stable. We will understand later in the presentation. It should be non-inducing, reversible. Again, depending on the target, it could be reversible, it could be irreversible. Again, based on the target, you have to decide the durability. Of course, it should be non-teratogenic, non-mutagenic, and you should have a patentable opportunity. And then finally, it should be manufacturable. So in terms of, if I could talk about specific assays, if you see, all the discovery criteria should be met. Okay, it should be safe in animal models, and it should be in a way that we are able to develop a good formulation out of it. It should be stable because you need to prove the stability studies for each of the formulation which you develop. Okay, you have to do the trial sites, patient recruitment, dosing range, side effect profiles, long-term safety, carcinogenicity studies. So these are all the things which gets into, like you start with the 10 molecules. By the time you make your molecules qualify for each of this criteria or parameters, finally you actually come up with only one drug which is ready for launch. Okay, so that's why it takes so long and so much of an effort to bring one successful drug into the market. So there has been a paradigm shift in drug screening. I would not like to name the company, but majority of the company during those days when I started my career as a DMPK scientist a couple of decades ago, the approach was in vivo testing used to be done, followed by in vitro testing. In other words, chemist will come to me with a 30 mg or 20 mg of compound, and he will ask me to test it directly into the animal model, which would be rats or mice based on the program specific models. And we used to prepare a formulation, dose it to the animals, take the samples, put it for bioanalysis, and come up with a plasma versus time concentration curve. And then used to calculate different PK parameters. And you won't believe, 9 out of 10 times, we used to end up having low viability, or in fact zero viability. And then we used to reverse pharmacology to understand that why we are not seeing the viability. Then we used to do the in vitro or testing, like is it a solubility issue, is it a permeability, or is it a metabolism issue? And we used to do this whole reverse pharmacology. But that was a scenario 2 decades ago. But now, within the last 2 decades, things have changed drastically in the industry across country and across world. And now you have all the in silico screening in process, which is in place. And after in silico screening, which is just using computer simulated models, we screen the molecules, we do docking studies, and then only selected scaffold comes out of it, which actually then gets into the chemistry synthesis, and then we do the in vitro profiling followed by in vivo testing. So earlier slide which I was talking about, that when you're talking about PK/ADME properties and safety liabilities, if they're tested quite early in drug discovery and development, then there is a chance that we can say fail early, fail cheap. It doesn't mean that we want a molecule to fail, okay? But the idea here is if you can identify those molecules which can fail early, then there is every chance that they fail cheap, so that later on you don't have to really spend more money on those. So that's why this extensive, aggressive screening on ADME and initial safety is very important in drug discovery and development. Okay, so let's shift the gear and get into few of the very basic in vitro ADME assays which are generally done, and for research scholars, I think it will be useful. So let's see the fate of a drug. When you take an oral pill through gut, and then it will either get excreted, it gets absorbed, and then through liver, it gets metabolized. You'll have the free drug which is available for the site of action to elicit the pharmacodynamic effect. There will be some drug which will be bound to the proteins. Liver will metabolize your drugs into metabolites. Now they are highly polar, so through kidney they get excreted. The drug which is unabsorbed will also get excreted through there. So this is the overall ADME which is going on. Actually, as we used to study pharmacology, like what body drugs or what body does to the drug is PK, right? It absorbs, it distributes, it metabolizes, and it excretes your drug. And what drug does to the body in terms of pharmaceutical pharmacodynamic effect, that's called pharmacodynamics. So let's talk about absorption, right? So absorption, as we all know, the transfer of drug from the site of administration into the bloodstream. Okay, so it will definitely depend on the rate of absorption and the fraction of the dose which has been administered. What are the factors? So like lipid solubility, how big is the molecule, what is the polarity, what is the route of administration. So all these factors will govern the absorption. When you talk of distribution, then again lipid solubility, the ionization potential, pKa value, extent of binding to the plasma, differences in the regional blood flows will define the distribution of a drug, that is from circulation to tissues or finite to the site of action. Metabolism, again, purely, of course, it can happen anywhere in intestine and liver, we'll see in later slides, but in general, either phase 1 or phase 2, what you're doing is actually making an active drug into an inactive metabolite, or you're converting an active drug into an active product, like is the case for phenacetin to acetaminophen, or you are just changing an inactive drug into an enhanced activity or an active drug, right, through biotransformation, which is metabolism. So, as all of you are very well aware, related reactions, be it oxidation, reduction, or even hydrolysis, versus you're working on phase 2 reactions where you're adding a moiety to the existing molecule and making it bulky, thereby more polar, so that it can easily excrete out. It could be glucuronide, it could be conjugation by sulfation, or acetylation. So all those things, finally, you are actually doing, helping the drug to become more polar in order to get excreted finally, right? So excretion, again, as I said, major route would be through kidneys. So what do we really want to learn out of this? So I would say from the preclinical ADME or the initial assays, we should be learning about the safe and tolerable exposure of a drug. Of course, we come up with a dose and a dose regimen, and we have a fair understanding of the ADME principles, all right? And then later on, we'll cover these few parameters which I feel are of more relevance to PK interpretation when you're doing initial studies. Okay, and then we will be talking about all these initial assays, especially for PG scholar students, that these are important assays, very simple assays to do, but this can give you a lot of understanding on the properties of a molecule from ADME perspective. All right, so let's get started quickly with aqueous solubility. So solubility, again, in a very simplest term, I would say it's a maximum amount of a chemical or a solute in a given volume of a solvent. Okay, so why it is important? Because it is a prerequisite. All of you are aware about metabolic stability, reaction phenotyping, CYP inhibition studies or induction studies. So all this data would be completely erroneous if you don't know the solubility of your molecule. Okay. At the same time, if you have solubility concerns, suppose it's an intrinsic property of a molecule that the solubility is very low, then whatever in vitro assays, like you are doing an artificial membrane permeability assays or canine adenocarcinoma cell line permeability study, or even a protein binding, there is very least chances that you will be able to detect any drug because the solubility is very low. So that's the importance of solubility. It is very important for the drug absorption, okay, drug dissolution, finally which leads to the drug permeation after oral administration. That's why aqueous solubility of your molecule of interest is very important. I'll spend a minute on this slide because this is very important for all the students to understand. So this is the classification, as you all know, that there are 4 kinds: class 1, 2, 3, and 4. High solubility, high permeability, low solubility, high permeability, high solubility, low permeability, and the 4th class is low solubility and low permeability. Now, no matter how we all wish that if our molecule falls in this category, okay, but the reality is above 90% of the molecules which are under development, unfortunately, they fall in this 2 range, mostly in this range or this range. So now that gives you an initial challenge to work with the formulation. So once you don't have a good solubility, you're going to struggle with every assay which you do initially for the drug development. So basically, as I was mentioning, with low solubility, of course, you're going to have a very least bioavailability. Dissolution would be very less. Finally, it will affect your permeability. But with our more understanding on ADME over a couple of decades or more than that, I think now we have good understanding. Even if your molecule is falling under class 2, which is low solubility, there are ways like you can reduce the particle size, you can form a soluble salt, you can add some surfactants, you can come up with the nanoparticles, or you can do some pH adjustments, or you can do a solid dispersion technique, and you can make your molecule or a drug more soluble. If your drug of interest falls in the 3rd category, which is low permeability is an issue, then nowadays there are absorption enhancing excipients, okay? There are transporters which plays a major role for low permeability. You can use efflux inhibitors, that is transporter inhibitors like verapamil, and many other inhibitors are there well known, which can be used in order to increase the permeability of your drug. And then lipid-filled capsules are also available. So here you can increase your low permeability. Suppose your molecule is very highly potent, like micromolar, I mean not micromolar, I would say picomole or a nanomole potent molecule, and it is having very low solubility, it is having very low permeability, then the way to go forward would be you can develop that into a prodrug, or you can work out with some surfactants, you can use liposomes, nanoparticles, because nanotechnology also has come into play nowadays, and that's why even if a drug falls under class 4 category, there is every chance that we can still develop that drug further. Okay, so that's about solubility. Again, all of you might have heard about kinetic and thermodynamic solubility. Very quickly, from an industry perspective, I just kept this if you do a kinetic, you can start from DMSO stock, all right? If you're doing a thermodynamic, that generally starts as a solid material. So the disadvantage here is not disadvantage, but this is how thermodynamic solubility is done. You need long mixing hours. Okay, it is kept for days together to achieve that equilibration. Okay, so control temperature, of course, you should have control temperature conditions. And eventually we aim to have a 10 mg per ml of target solubility. Okay. But the throughput is very low. As you can understand, minimum 24 hours is required for mixing. That's why it is only 20 compounds a day we can screen if you are planning to do a thermodynamic solubility. When you talk of kinetic solubility, we start with the DMSO stock. So the advantage is you get a clear solution at first go. Now you can do various incubations. You can use different temperatures. You can use solubility of 100 microgram per ml, but you can do it in a plate format, in a 96 well plate format. So the advantage is up to 150 compounds a day can be screened. So that's the difference, but both are equally accepted. I would suggest that it's better to do kinetic because this is quick and it gives you an early indication if there is any solubility issue with your drug. So how it is being done, a very simple assay I have mentioned here. Very quickly, I will say, just take your drug. You should have some idea about what amount is needed to saturate a volume of water. Just take 5 times of that quantity. Take it in 3 different in vessels. Maintain at a particular temperature. Allow it to equilibrate. After 24 hours, you can take out 1 vessel, and then you can do MSMS or an HPLC analysis and just see what is the concentration you are getting. Okay, and then after 24 hours, another vessel, and then third vessel can be taken. So whenever your concentration which you're measuring in the last 2 vessels do not differ by more than 15%, means you have reached the equilibration, and that solubility is the equilibrium solubility you're looking for. Okay, so it's a pretty straightforward assay. Octanol solubility, again, octanol is nothing but it is octane 1-ol. It's an organic compound, and very quickly, I would just mention that basically octanol and water both are immiscible. So together, it gives you an impression of our gut wall, which is a phospholipid membrane. So that's the principle behind octanol solubility. You can very well use. You might have heard about a term called it is called as log P. So for finding out log P, octanol water partition coefficient assay is being used. Very simple assay to do. Take equal volume of water, take octanol, dissolve your molecule into water, mix it well, equal amount of water to that. You have to add equal volume of octanol, stir it well, keep it for equilibration. After a desired period, take it out, centrifuge, separate the layers, look into the volume of, concentration into octanol layer versus concentration into water layer, and that's all. So this is, again, this will give you a very good information, early indication on if there are any permeability issues. Okay, let's talk quickly about plasma stability assays. So now, many a times I see students, especially masters, when they do dissertation work and they choose a good drug, but there is a huge difference between they collect the samples versus when they actually do the analysis, and then they try to correlate the data which they obtain versus literature reported PK parameters. It doesn't match, but it could be as simple as a stability issue in the plasma itself. So I would always suggest that please do a plasma stability assay, which is very simple. It can be done within your lab with very limited resources what you have, and it will give you very good information, be it a new molecule or a known molecule. Okay, why should we do that? So this is the reason, because as you all know, in blood there are hydrolytic enzymes, cholinesterase, lipase, dehydropeptidase, alkaline phosphatase, aldolase. So what happens is, if your molecule is a substrate for any of these enzymes, it's going to cleave that. So your drug is no more stable in plasma anymore. So just an example I've taken here, like acetylcholinesterase, it has a very strict site which is very labile. So what happens is it undergoes hydrolysis, and now you have acetic acid and choline once it is exposed to blood. So whenever your molecule, if you're looking at the structure, if you see an ester, amide, carbamide, lactam, for antibiotics, lactone or sulfonamide. These are all the very labile groups for plasma degradation. So you may say that ester amides are very common, but that's fine. Even if it is there, sometimes they're masked, so it may not be subjected to degradation. But if you have at least these structures, please do plasma degradation very early. All right. So again, for prodrugs, it is like one of the very important, very important aspects because many times drugs are developed in a way that the moment it is coming in contact with blood, it is absorbed, it will convert it into its active form, right? So that needs to be watched very closely. So any hydrolysis options are there for your product, then you can understand it very well, whether your concept works or not, simply by doing a plasma stability assay, right? So again, for anti-drug, which are soft drugs, which is opposite of prodrug, there again, they are locally active, and then later on you want to make it inactive once it is into bloodstream, even that can be done if they have a hydrolytic site within them. Okay, so now you understood the importance of plasma stability. All right, so diagnosis poor in vivo performance, it will, any liabilities upfront you will come to know. Compounds in vivo studies, at least you will come to know whether you need to stabilize your plasma samples after the PK study or efficacy study or not, right? How do we do it? Again, very simple. Plasma is diluted with phosphate buffer at a room temperature, 37 degrees. Any drug concentration, we used to do it at 200 micromolar concentration. Incubate it at different time points, take out the sample, denature the protein using isoniazid, acetonitrile or methanol, and then subject your supernatant for HPLC or mass spec analysis. And then you can very well find out the plasma stability for your compound. Okay. Very quickly, let's get into, we have covered very few basics on solubility, octanol, plasma stability. Now, if an oral drug, if you oral pill you're taking, it goes through your stomach into the intestine. Okay. So even before getting into an in vivo assay, you can do a simulated gastric fluid or intestinal fluid stability. All right. And it's like non-enzymatic reactions or processes. If you want to track, then you don't have to wait for a drug to be absorbed. In stomach itself, you can check that. So these are all the tools which helps you whether your molecule can go further or not, or if you need any modification. So what happens is, if at all a drug is not stable at acidic pH, you know that in stomach it's going to degrade. Or if it is not stable at basic pH, then in intestine it may degrade. So that's the reason you can do a simulated gastric fluid. Again, very simple assay with minimal things in your lab. You can do it. You can adjust the pH to acidic, add a pepsine, it becomes a simulated gastric fluid. If you want to do a simulated insulin, just add pancreatine and you can do a intestinal fluid stability within that. And very quick assay at different time points within 1 day you can complete. And this can also give you some insight into how your molecule will be impacted when actual formulation you're going to administer into preclinical species. All right. So microsomal stability, again, I won't spend much time on it because all of you are very well aware of it, but please understand, very easy to do microsomal stability, and for any program, irrespective of the projects you may be working on, be it anti-inflammatory, be it diabetic projects or an oncology or any projects you're working on, have your microsomal stability as one of the initial liability assay for screening of your molecules, and it will help. Easy assay, quick assay, and gives you any metabolism related liabilities are there on your molecule, you can detect that very early. Okay. I don't want to get into the detailed procedure, but I'll be sharing the presentation. But I just mentioned this because many of you are veterinarians. So within your lab, you can actually prepare microsomes very well. So that's why I just mentioned it, that you can, from a rat or a mouse, you can easily prepare microsomes with very basic facilities. Of course, here you need an ultracentrifuge, which can go up to 1 lakh G. But apart from that, rest all the things are very simple, and you can develop your own microsomes within the lab. Okay. This is the procedure of how exactly it is being prepared, how you really isolate, and then you can actually do a stability assay, right? So stability assay, again, later on we'll be talking about it, but it's very simple to do. You have to just need your microsomes, the phosphate buffer, NADPH, which is going to be a cofactor, and your drug. So the moment you add NADPH, your reaction is going to start, and then after 15 minutes, 30 minutes, 1 hour, 2 hour, you can take out the sample and freeze your reaction by using acetonitrile, and then just see the disappearance of your parent drug. So I start with, say, 100 microgram, 100 micromolar. After 1 hour, if I'm left with 50 micromolar, that's the typical half-life understanding in vitro. It can give you about your microsomal stability. So intrinsic clearance you can very well find from this. So let's move on. Let's talk about permeability now. So you see, many times chemists, they don't really understand the difference between a chemical structure and the disconnect in the overall bioavailability. But as you guys are going to be a DMPK scientist, you will definitely understand you should understand that how this chemical structure is not really reflecting same in terms of good ADME properties, why you're having low bioavailabilities. So I want all of you to really understand the permeability concept as well. Okay, so let's just see if from a gut human, if you see the drug actually gets absorbed through gut wall, okay, then through portal vein, it's going to reach out to liver, which is a site of metabolism. At the same time, elimination also starts, and then through systemic, it reaches to the different parts of the body. All right. So bioavailability is not only one aspect. Actually, it covers from gut permeability all the way to metabolism. Everything will decide the importance of your bioavailability of your molecule. So it's just not a function of only one mechanism. There are several mechanisms involved, and that's why I feel that you should understand all these things. All right. So when you're taking a drug, basically through stomach and intestine, it is release plus dissolution. So solubility plus dissolution. From the gut wall, it is formation, and from the elimination, it is generally through metabolism, efflux, because in your gut wall, there are efflux transporters, which actually efflux your drug there itself. You may still get low bioavailability. And finally, you will have metabolism through liver. So all these things will be deciding the bioavailability of your molecule. So very quickly, let us understand our lipid gut wall membrane structure. Right? All of you know about it. It's a sandwich model, hydrophilic regions. Sandwiching the hydrophobic region. You got transmembrane proteins, you got glycoproteins. So this is the typical structure. So when I just take intestinal mucosa, and now if I just take this section, if you see there are going to be apical side, there is a basolateral side. So when a transport of drug through intestine I'm talking about, there could be only few scenarios. Either the transport of the drug is transcellular, which is the most common way of transport of any drug through the intestinal barrier is transcellular. It can be paracellular, or it could be carrier mediated. All right. So again, through endocytosis also it happens, and there are certain mechanisms called as anti-transport mechanism. Like metabolism happens, then drug will be transported back, or there could be other mechanisms involved as well. So this is the whole concept. Talking about on this slide, passive diffusion. So you can see molecules like water or other compounds and the small lipids. Basically, when it is a passive diffusion, this is always over the concentration gradient. So from higher concentration gradient to lower, that's how it transports. When you're talking of facilitated diffusion, what happens is the carrier protein will change the shape of the transmembrane protein itself, and that will be the way for entry and exit. So that becomes your diffusion. When you're talking of an active transport, here, if you can see, there is no concentration gradient in active transport, but instead there is an energy, ATP involved. And here you can see now, I would say in last decade or so, a lot of understanding has come about all the transporters, different transporters available. So this is all about role of transporters as well in your permeability. So very quickly to summarize, passive.
Dr. Deepak, you are not audible.
Hello?
Yeah, now it's fine. Yeah. Thank you.
I'm so sorry. Maybe the charging for the AirPods just went down. I'm so sorry. All right. Is it okay now? Is it? Am I audible now?
It's clear. It's clear. Please carry on.
All right. So let's quickly let me summarize the passive diffusion just for the understanding of the students. It could be transcellular. It could be paracellular. Okay. And then log the molecular weight of your molecule, polar surface area of that. This will all define the permeability in a passive diffusion. Whenever it is a carrier mediated, it could be facilitated transport, okay, through membrane bound proteins, or it would be concentration gradient but no energy. When it's an active transport, it is always energy dependent, okay. And then there's receptor mediated drug carrier protein forms a complex, and then through pinocytosis also it can happen. All right. And anti-transport, as I was talking about, it requires energy. And efflux transporters also will be anti-transport because it will not allow your drug to get absorbed, and it will transport it back into the lumen. All right. And so a passive, as you can clearly see in this, it's a rate of drug absorption versus concentration function. Okay. So that's how it appears like this, whereas active and carrier mediated is going to take some time, and then it reaches the plateau, and then over a period of time it happens. All right. So what are the factors? Let's understand very quickly the factors which are responsible for drug absorption. So I'll just name it, won't get into much details of it, but as I just mentioned, surface area, intestine is always a better option for absorption than stomach because you've got huge surface area. Blood flow also is huge there. So again, more chances of getting absorption. Gastric emptying time, all of you know why we take pills after lunch and before lunch and all those things. So this really depends on that. Drug transporters, again, plays a very major role, and your understanding on drug transporters is very important. From a physicochemical properties point of view, we already talked about solubility and all those things, ionization, pKa, partitioning between, already we have discussed polymorphism. This is not a genetic polymorphism. We are talking about polymorphism of your drug, of your formulation. Okay. So sometimes the different polymorphs will have a different physicochemical properties, particle size, complexation. So all this will affect your absorption. So let's talk about what are the models available. Okay, so there are biological systems, there are non-biological systems, and there are in silico techniques also. So very quickly through pictorial, this thing you can see, we used to just do using a chamber many years ago. It is a very cumbersome process where we used to take the intestinal membrane and then parts of it. We used to take it in a device, and then it used to take a long time. And with time, a lot of variability used to come. But again, one of the method which was earlier used was this. You have now the most popular method is for many years now is Keiko2 cell lines, right? So it also expresses some of the metabolizing enzymes, not all of them, but it expresses the transporters. So that's a wonderful model for a permeability. Suppose you don't want to use a cell-based assay initially, then you can use immobilized artificial membrane columns, or even going forward, a PEMPA assay or an in silico assay. So I'll quickly talk about pros and cons, just one pros and cons for each of these assays. Like permeability is monitored by measuring of disappearance of the compound from the donor side or the appearance on the acceptor side. So that's an excised tissue assay, right? So various parts of the gut can be used, and you can compare the absorption in different regions within the intestine. That's a great thing. But there is a variability in the tissue viability between assay setup, and with time, the tissue physiology slightly changes. So that also impacts your results. Cell-based assay, monolayers, fantastic monolayer confluent layer you can get. Automation is also possible. Any in vivo gut characteristic can be really expressed and used for this cell-based assays. As I'm saying, not only CACO2, you have MDCK, H229, other cell lines also which can be used. So it's a great model. Both ways direction you can check, apical to basolateral and basolateral to apical. Both monolayer you can check. Even the role of a PGP substrate, because suppose your drug is a PGP substrate or not, you can come to know if you incorporate a PGP inhibitor like verapamil in your assay. All right. So this is typically just an example I have shown, that when you use without verapamil and with verapamil, see the difference in the concentration from B to A to A to B. See the difference. So this establishes the role of PGP substrate affinity for your molecule. So again, very high expression of P glycoproteins, but low expressions of metabolizing enzymes. So inter-experiment, inter-laboratory variabilities are there because it is cell-based assay. But still, as on date, across country, across world, CACO2 is a widely accepted model for studying permeability studies. Artificial membrane, as I said, as the name suggests, it doesn't have a live cell into it. I mean, no cells are involved, but they use a device is there, and they use a membrane within that, okay, which will try to mimic a phospholipid membrane. So it could be PEMPA, it could be a column base, both are there early in drug discovery, can be used. This is typically how it is being done. The composition, we have come a long way. There was a time when phospholipid solvated into dodecaine was used as a solution in PEMPA, and nowadays formulation of lipids trying to mimic exactly like a gut membrane is being used. Okay, so general requirement for permeability assay, whenever you do this assay, just have your controls. You should have a high oral absorption, low oral absorption, and one which is a substrate of an efflux transporter. Always include these three, and I think you are good to go. Very quickly, in silico nowadays, as I'm saying, even before you get into in vitro, a lot of work can be done on in silico. If you have the structure, if you have your crystal structure ready with you, or even a 3D structure available with you, there are many softwares available which can tell you the liabilities early in the drug discovery through in silico method. So basically, molecular weight, log P, hydrogen bond donor, hydrogen bond acceptor, solubility. So whether your molecule qualifies for Lipinski's rule of 5 or not, we can very well establish it before even getting inside your lab. So that's the advantage of doing in silico method. But again, in silico methods is good, but there is huge inter-individual variability in drug response, inter-laboratory variations in experimental protocols. So again, the predictability is not that great, but still, it's like you have thousands of molecules. At least it will help you to screen and get back to a narrow part of the funnel through in silico method. Okay, so this is in summary for your permeability assays. I would just say that overall, this is your permeability. Always remember passive diffusion and active transport. From passive diffusion, transcellular diffusion, you can definitely use PEMPA, immobilized artificial membrane HPLC column. You can use cell-based for your paracellular. For active uptake, you can do CACO2. For PGP role, again, you can use CACO2. All right. So let me now quickly get into PK properties. Like all of you are PK experts, and I just wanted to give you a perspective from screening. I mean, we have worked on several projects, and I'm just going to give you gist of few parameters which will really help you decide the fate of your molecule. So that's why what are those key parameters, PK parameters, how they're interrelated, and how do we interpret the preclinical PK parameters. All right. So let's just understand again. We already talked about it. There was a time when our understanding on PK, I mean, the attrition of due to PK was more than 40%. And now it is as low as 7% because of our improved understanding. Okay. So what is the role of DMPK scientists? So is it that we should always go behind a medicinal chemist, or is it that a pharmacology scientist to understand? Because you as a scientist, you have to understand that you are right in the center. Okay, when a chemist is actually, every chemist feels that they have come up with the best molecule in the world. So any data which you give back to them and which is not as per their expectation, they're always going to be unhappy about it. Okay, a biologist who established that your drug is highly potent, the moment you say that it's not even bioavailable, it is not even available at the site of action as a DMPK scientist, biologist is not going to be happy. Okay, so I would say you are sandwiched as a DMPK scientist. So I would just say that instead of choosing this, all of the above is the option for you. So you have to work closely with chemist to help him work on getting rid of the liabilities of DMPK early from his structure. With a biologist, again, you can make him understand the difference between an in vitro biology assay versus an actual in vivo experiment for ADME properties, and why there is no drug available and why we are not seeing the efficacy. So this is what I call it as through this. So pharmacokinetic. Hello, is my voice audible?
Yeah, Mr. Ravi Prakash, please mute your audio.
Yeah, some echo is coming actually. Yeah.
Yeah, somebody is unmuting it. So I'm just.
Okay. So all right. So pharmacokinetics, dynamics, all of you very well understand now, at least by now, that this is what it talks about, like the main drug versus metabolite, sigmoidal for your Emax to your what drug does to the body. We have talked about this, but I will just give you a different perspective from a just not for this perspective for your understanding. We are already let's see, systemic circulation, drug can actually either be excreted after metabolism, after getting metabolism, or it can directly get excreted. So the process of excretion after metabolism, the parameter which is important here is clearance. When we say we have a tissue distribution, that is what we call it as volume of distribution. So that parameter comes into the play. You're getting the point. So elimination is nothing but metabolism plus excretion, and disposition is distribution plus elimination. Okay. So what I say is primary PK parameters for screening process is clearance and volume of distribution, and secondary parameters are half-life and bioavailability. Okay. So let's very quickly in a couple of slides we'll go through each of these parameters. Let's start with clearance. So it actually describes how efficiently or rapidly is drug actually is eliminating from the body. Okay. So elimination, the moment I say it is function of metabolism. Metabolism can again happen in liver, it can happen in intestine, it can happen in lung, it can even happen in kidney. Excretion can be apart from urine, through bile, through saliva, through milk. These are all the ways, different ways through which it can get eliminated. So basically, as you all know, rate of elimination versus the concentration in the blood is what we define it as clearance. But this I just want to emphasize, especially for the students. See, clearance, I see many theses, many research dissertation work, when students come, they tend to report clearance after oral route as well. You can actually, but please understand very basic difference that when you're doing an intravenous study, the assumption is that 100% is available in the systemic circulation. So after that, when you divide your dose by AUC, you actually get an accurate clearance. Okay. But when you're doing an oral dosing and you don't have an idea about the bioavailability, you actually don't know how much drug is there to start with in your systemic circulation. So your clearance assumptions can be slightly off. So if at all you want to report after an oral route your clearance, please make sure that you keep your bioavailability into the play. And how do you do that? Is just clearance when it's same as IV, but you have to just divide it by your bioavailability. So that gives you an accurate clearance even after PO dose. So just remember this small things. What is the impact? You can clearly see. The one which I'm highlighting here in the green, when you have a, suppose let's take this red one where the 10 ml per minute, which is higher clearance, you can just see how fast it is going out. So once it is 5-fold decrease in clearance, you just see how long it is going to stay in your body. So basically, it's taking long time. So what is the parameter it is correlated to? Straight away, it is your half-life. So longer the clearance, I mean the slower the clearance, your half-life is going to be longer. Your faster the clearance, your half-life is going to be short. So the parameter which is directly related to clearance is always your T-half, proportionally, okay? Same way, your AUC would also be higher. I'm giving you just few inputs because when you read some data, you should be able to interpret out of it. So that's what, whenever you see higher clearance, always remember the half-life and AUC are always going to be slightly less. When you have a longer clearance, slow clearance, both half-life as well as AUC would be higher. So this is the relationship between the three parameters. So when you're talking about different species, and let me talk about extraction ratios, okay, from liver. So what happens is, suppose a systemic blood clearance is known. This is just an example in cyno, it is 25 ml, suppose it is there. So now if I have to calculate a hepatic ratio, extraction ratio in cyno, what you need to do is just take this clearance, divide it by the blood flow in the liver. So I see the blood flow in the liver for cyno is 44. So I just divide this by 44, I get 0.56. So this is an intermediate clearance. Why? Because this is the thumb rule. So thumb rule is whenever you see clearance, low clearance, the extraction ratio, if you calculate, it is going to be less than 0.3. Whenever you see an extraction ratio of higher than 0.7, means it is always going to be high clearance. You see, so this is just an idea about just from a clearance, you can actually predict the hepatic extraction ratios, and you can find out where your molecule is going to lie in terms of clearance. How do you arrive at total clearance? Of course, it's going to be hepatic and renal. Of course, towards the later part of my presentation, I'll tell you that it's not only this two, there is going to be another factor added up also, but let's consider hepatic and renal. So when you consider hepatic clearance and renal clearance, the total comes via total clearance of your molecule. So when you talk of in vitro microsomes, hepatic turnover, this is how through IVIVC, we try to correlate the in vivo clearance. So that's where this particular clearance concept comes into the play. Let's move on to volume of distribution. Okay, so I won't take much time on it, but let me tell you volume of distribution. We always say apparent volume of distribution. So for students, just want to make you understand, it's very important to understand it is actually apparent volume. All right. So basically, all of you know that volume of distribution is nothing but an amount in body versus blood or plasma concentrations. So why do we call it as apparent? Because actually it rarely reflects a true physiologic volume. That's why we call it as apparent. Let's understand by this simple example. You just have this container. You have 10 ml of water, 10 liters of water inside. You add 5 mg of drug inside. Okay. Now, if I just back calculate, so how much is the volume? I have to just divide amount by concentration. So 5 mg divided by 0.5, I get 10 liters. So this is 10 liters of volume. Now, say for example, hypothetically, you consider as in our body, apart from fluids, there are so many organs. Let's consider we're adding beads into the same container. What happens? The drug will get absorbed to these beads. So now in your solution, the concentration is going to go down. Say for example, it is 0.05 mg. Now again, I go back and calculate what is the volume in beaker. See amount by concentration, it comes to 100 liters. Although we know it's going to be 10 liters, it shows 100 liters. So that's why we call it as apparent volume. Okay. So same beaker, the calculated volume could be different because a lot of organs, a lot of other things are there in play. So that's why we call volume of distribution as apparent volume of distribution. Okay. If you ask me the correlation, so it correlates well with the body weight and the similar volumes for preclinical species. Okay. So this you will understand from this factor what I'm trying to explain you. So you see this difference. You have this one you have got 5 liters per kg volume of distribution. Here it is 1 liter. From the graph itself, it tells you what is the factor which it is going to affect. it is going to affect your T half, because the T half is going to increase. Here it's going to be more, here it is going to be less. Okay, so what happens is your T half will change drastically with your volume of distribution. So when you are looking at volume of distribution, a higher volume of distribution, you can have some idea that there will be an increase in T half as well. But please understand, I have categorically I have mentioned that it may not lead to change in your AUC. Okay, because although it looks like your graph is like this, but when you compare it with a higher concentration initially, here if I form a trapezoid, there is every chance that AUC for this may come more compared to all the summation of trapezoid areas of this. So volume of distribution may not lead to change in AUC, but it does lead to change in T half. Okay. So again, for there also there are values in place. Volume of distribution is steady state. Whenever it is 0.3, less than 0.3, we say it is small volume of distribution. Whenever it is more than 0.7 liters per kg, we call it as high volume of distribution. Where low volume of distribution is useful for vascular or extravascular targets. Where high volume of distribution for intracellular targets. So you can see where we want more of a tissue distribution, intracellular target, it is high volume of distribution required. That's about VD. Let's quickly talk about half-life. All of you are expert in that. We already talked about it. Higher the volume of distribution, higher is going to be your T half. We have learned already, lower the clearance, it is inversely proportional. So higher is going to be a T half. So why I'm just emphasizing is whenever you look at this paper or any numbers, try to interpret, okay? Look at these parameters and try to understand and draw some conclusions out of it. All right. So half-life, all of you must know very well by now, because half-life, shorter half-lives are required than daily dosing. Okay, you have once a day dosing, then you would wish that at least 10 hours or 8 hours above half-life would be required. So based on what is your clinical outcome, the half-life can give you an idea about what dosage regimen or the duration you want to keep. Please understand here, in this scenario, I've just taken hypothetical example. Of course, these are real examples, but I've not put the names. See, the efficacious dose is 100 nanomolars, okay, 100 nanogram per ml. Now, these two compounds are there. For both, the half-life is same, but you cannot really choose purely based on the half-life, because here, the concentration is way below your efficacious dose. So, you always have to keep into mind, blindly, we cannot just take a call or drop a molecule purely based on a half-life. You should keep into perspective other like AUC, like clearance, or other factors also plays a role when you decide which molecule is better than the other during initial screening and admin. Okay. Quickly about bioavailability. Now I think the same slide I'm showing here, you will very well understand what I mean by bioavailability now, because these are all the factors which plays into the role. So again, oral bioavailability versus IV, we know the formula. We have to do it by 2 routes. Only then you get a bioavailability for the other route. It could be oral, it could be any other route, but the one route is fixed, which is by systemic route. You have to do one study at least. Different formulations, you can have a bioavailability, like BAB studies, we say, between a tablet, between a solution, you can still do this. It clearly tells you between a solution versus a suspension, right? So of course, solution is going to have a higher bioavailability compared to suspension if your bioavailability is only limited by permeability. Okay. Protein binding, again, very simple assay to do. Of course, not that simple because analysis is little difficult here, but very initial assay which can be done in a lab. As we know, the hypothesis, it's always unbound drug responsible for the site of action, for eliciting the desired action. Where does your drug bound? It gets to albumin, glycoproteins, lipoproteins. So when you are looking for, also the point I was trying to tell you here is protein binding also plays an important role. Slight change in protein binding will lead to a lot of change in the PK properties. Okay, so please pay attention to protein binding as well. And finally, for multiple dose regimens, you have to have a balance, like what you're looking for. Is it a clearance? It is a half-life, or what is a efficacious concentration? What is your final destination or the objective for a molecule or for a disease target. Based on that, it can be done. Okay. So for the PK parameters, quickly in summary, as you all know, the primary parameters for optimization during early drug discovery in preclinical phase, always going to be clearance, volume of distribution, half-life, oral bioavailability, AUCs, right? So any small structural change will have impact on any of these parameters. Why I emphasize this much is because whenever with this, because chemists will never know the importance of clearance or a volume of distribution or a AUC or a bioavailability. A bioavailability, of course, they will understand because this is the only figure they understand. But you have to go back and talk to them, make them understand that, okay, if you can do some slight change in your structures, maybe we can slightly change this and eventually bioavailability can be improved. All right. So very quickly. So Dr. Srinivas, how much time I have? Can you just tell me so accordingly I can.
Yeah, Dr. Deepak, you may how many slides till to go? 10, 15 minutes can we have?
Yeah. In less than 15 minutes, I will conclude. Yeah.
Yeah, please. Thank you.
All right. So since many of you are veterinarians, many of you are pharmacy students who have done in and out animal experimentations, just wanted to touch base on few of those points which are really important to this scenario, that all of us should really ask ourselves, is it really required? Are there any better alternatives? Can we really do drug discovery without animal experimentation? In silico methods of testing versus in vitro versus animal experimentation. So I would just I would like to show you this, of course, there are huge variabilities across species, okay? So you just can't predict. Of course, there's allometric scaling, there is IVC, all the prediction is possible, but we are not yet there, okay? So I'll go back to this slide and I will say my answer to this is, of course, there are better alternatives, but is it really possible without drug discovery? I mean, without animal experimentation? Answer is no. We just can't do that. You can do as much in silico as possible. We can do as many in vitro assays as possible, but still, eventually, you have to get into animal experimentation and generate that much required data in a live biological system. So it's an amalgamation of all these things. Of course, we can reduce the usage of animals undoubtedly, but we have to do animal studies. Just wanted to give my perspective on that. Okay. So let me just, since I talked about alternatives, very quickly I'll take you through a few more options in ADME on hepatocytes and enterocytes as well. Okay. So of course, this is the main goal of accurate assessment of human drug properties. Basically, my association is with a company called In Vitro ADMET Labs, which are involved in providing solutions purely for hepatic and enteric technologies. I thought few tools I will share here which will be useful for the students. Okay, so let's just I think now all of you know better than me about this slide. So let's just understand. I'm going to focus majorly on metabolism here. Okay, so from the gut wall, when it gets into liver, it gets to metabolism. All of us know that. What I'm going to emphasize, and first I'll talk about liver, and then I will talk about the intestine. Okay, because what we all need to understand as a scientist is that many metabolism happens in enteric walls as well, intestinal wall as well. Okay, so that's what I want to understand, and I'll just talk about few technologies which can do both. All right. So here's the concept. Why intestine? Because 3A4, which is responsible for metabolism of more than 70% of the drugs available over the counter, the metabolism, the isoform which is responsible is CYP3A4, and that is expressed heavily in intestine as well. All these years we were considering that it is there in liver, but no, it's expressed in intestine as well. That's why we say that you have to study enteric metabolism as well, apart from hepatic metabolism. Okay, so let's talk about hepatocytes. So for students, please learn this. This is the gold standard. If anybody asks you what is the gold standard to study the metabolism,
always say it is hepatocyte. Even now it is. Of course, there are newer technologies which has come up, but by far most acceptable assay across globe is hepatocytes for metabolism studies. Why? In a cryopreserved hepatocyte, why do we want to? Why? So the reason is because you can schedule your experiments, you can repeat the experiments, you can use pre-characterized hepatocyte. Okay, you can use a pool hepatocyte, which is a multiple donor, so that any variability can be captured early in drug discovery. So all these options are now available, which were not there earlier, couple of decades ago. So that's why hepatocytes is the gold standard. So you can use cryopreserved hepatocytes early in drug discovery. So of course, cryopreserved comes with a recovery medium because it is in a cryopreserved state. You have to thaw that, so you have to use universal cryopreserved recovery medium. It is a patented medium, and all it is characterized, the age of the person, gender, race, yield, viability. So generally yield is somewhere around 6.2 million cells. Viability is always more than 90. So nowadays you might have heard about the term, like earlier it used to be suspension. Now why do we say platable hepatocyte? So please friends understand that platable hepatocyte has its own advantage. Okay, in suspension actually it will lose the viability very soon. So very limited time you have to do your assays. Okay, so so for a longer assays, so for the longer assays you cannot use suspension hepatocyte. So you have to use platable hepatocyte. Okay, so where can you use? As I was mentioning, long term, you want to do hepatotoxicity, you want to do metabolism dependent drug toxicity, time dependent inhibition studies, enzyme induction, efflux transporter studies. All these 10 can be done using platable hepatocyte. This is how the hepatocyte looks like. I don't know how far the screen resolution is there, but they're beautiful across species. They are very well laid out, beautiful layers you can see of a platable cryopreserved hepatocyte. And in terms of expression of isoforms like 1A2 induction, you can use using omeprazole, phenacetin, rifampicin. You can use all of them very nicely. Like induction is very well expressed for 1A2. Even if you look for the expression of 2B6, these are different lots. 3A4, so you have lot specific, so you can choose. Suppose if I know my drug is getting metabolized by 3A4, just to boost up my screening assays, I can always look for a lot of a particular lot of hepatocytes where 3A4 is expressed very well, so that I can screen my molecules very easily, right? So this is the expression level. As I was telling you, more than 90% viability, 100% confluent. All the major CYP forms are very well expressed. Long durations you can use it, and then large numbers of vials are already there. So all the different animal models, all the way to human, you can use. Okay. What about the new hepatocyte? So re-cryopreserved. So this is something called as pooled cryopreserved. So I was talking about this, where I was talking about individual lots. As you can see, these are different of the same lot, different expressions. Now here I'm talking about pooled hepatocyte, because now let's talk of genetic polymorphism. All of you guys know that there is huge variability among individuals across globe, right? Somebody could be slow metabolizer, somebody could be high metabolizer. So how do we factor this variability early in our screening during ADME studies? This is what you need to do. Take a pooled hepatocyte. So you have different individuals, donors, liver, hepatocytes are mixed up. So now you can really minimize the individual differences. Even that is a patented product actually. As I was telling you, now you very well know suspension versus platable. Where all you can use, so you can use for metabolic stability, for metabolic profiling, reaction phenotyping. Reaction phenotyping is nothing but I will just be doing individual isoforms like 3A4 alone, 2D6, 2C9, 2C19. So individual forms I'll be using. Let's move on quickly into enterocytes. So I talked about hepatocytes. Okay. Now let's talk about enterocytes. So I'll go back, take you to 3A4. Okay, expression in intestine. So what is a key cell type for oral bioavailability? Why do I say that is major part of my presentation, I've talked about permeability. So what is that one place which we are targeting? That is intestine, right? So how can we forget and how can we not pay attention to the metabolism which may happen in enterocyte? That's why I say that for oral bioavailability, let's start paying attention to enterocytes as well. There could be intestinal drug-drug interaction because so many administered food is there, nutrients are there, that can all lead to metabolism within the intestine, and that can lead to the first effect, right? That's why enterocytes are important. So right now they are, of course, now they are commercially available. This is from last year onwards. Now they are commercially available as well. So let's quickly. So they are smaller. Okay, you might have seen hepatocytes. These are smaller, but yet beautiful they look. Mainly single cells, but they're smaller than hepatocytes for sure. In terms of protein content, as you can see, at least 5 times less protein in enterocytes compared to hepatocytes. How about the yield? Again, in yield, they are much less. So yield is also less, and the viability is also less compared to hepatocytes. Okay. So this is again a different. Exactly, I just mentioned about different lots. We do characterize enzymes as well, and for drug metabolizing enzymes, for uptake and efflux transporters, for enterocyte markers, all these things are in place, right? What are the cell-specific marker? How do we know? How do you differentiate? So, like sucrose isomaltase versus maltase glucoamylase, both MAGMAM and SI, they are actually highly expressed in enterocytes. But in hepatocytes, as you all know, it is albumin. So this marker gene can be used to really characterize the enterocyte hepatocytes. And so it's already been done. As you can see, clearly the enterocytes give you much more expression of marker gene for specific for the enterocytes and albumin for the hepatocytes. Okay, and I just want to want you guys to take it as a take-home message very quickly that tip 3A4 here onwards, please remember in intestine, out of all the enzymes being expressed, 82%. Okay, so where 3A4 is expressed more, it is in intestine. I don't say that stop doing hepatocyte studies, but please pay attention to enterocytes, to intestine as well. Okay, so again, this is a comparison of different isoforms. 2x1 actually specifically very specific point. Okay, again, viability and yield are lower, but still a better system to work on early in drug discovery. Okay, so entry applications. So as I said, for metabolic stability, and in one of my earlier slide, I was just telling that when you're looking for total clearance, so this is the factor I was saying that now that you know enteric metabolism, you can always add this factor, the enteric metabolism factor, also into your total clearance, right? So it is not only renal, it is not only hepatic, but it is intestinal. Total all the clearances. That's how you get the total clearance. So again, this is the most recent. It was launched in last quarter of last year. It's called 999 Elite. So as we say, Elite means more than 90% viability, more than 90% confluency. You can do more than 9 days of culture using this. Okay. So something called as METMAX. I thought it is worth sharing this. So that's why I've kept it, but I won't take much time, more than 30 seconds. So the only difference for METMAX versus a normal human hepatocyte is in METMAX hepatocytes, these are porous. So what has happened, let's understand from this. This is an intact hepatocyte when you're talking of hepatocyte, but we have made this thing porous. Why porous and what is the advantage? I'll explain you in a while. This is the advantage. You see, when you use microsomes, you only have endoplasmic reticulum, right? Related enzymes. When you use S9, you do have that, some portion of cytosol. When you use an intact hepatocyte, you have almost everything expressing there available. With METMAX also, everything is possible. Then what's the difference? Why should you use METMAX? This is the reason. Why many of the people don't use hepatocyte? Because it is not that straightforward. You need to have a liquid nitrogen, right? But microsomes I see most of the companies, most of the students tend to use early in drug discovery because it's easy, right? It's just a vial, put it in -80, take it out, thaw, take the required volume, start your assay, you're done in 2 hours. But it's not the case with hepatocytes. So with METMEX, now because we have made it porous, it is possible to store it at -80, right? You don't have to do centrifugation. You don't have to do any of this. So it is as good as using a microsome. Yet it will have all the characteristics of an hepatocyte. Okay. So that's the reason I say this new technology for students that please read about it. And I'll share quite a few many papers on this. We have published, and you can use METMEX very initially. Okay. For herbals, this is just for the enteric activity. We have checked for a 3A4 for different expression of 3A4, and you will be amazed to see that many herbal things actually tend to enhance the expression of 3A4. So it leads to inhibition or induction. So that's very important for herbal assays. Actually, all those people who are working with herbals, very simple, not simple assay, but at least in a lab you can do this assay. You should be able to do it if you want to screen your herbal drugs for the CYP3 activities. Okay. So this is the whole application of enteric thing. So in the summary, I just want to say that summary for hepatocytes and all, we always do animal hepatocyte studies. Then we try to extrapolate into animals. We do comparison in human, and from human through IV-IVC, we try to predict human in vivo characteristics. So that's how IV-IVC comes into the play. So what do we suggest now that time has come that apart from hepatocytes, do the same thing using cryopreserved intestinal mucosa as well. Okay. So now, as I said, towards the last part of it, in last 3-4 minutes, I will just, because no matter how good a DMPK scientist you are, if you don't understand bioanalysis, then I think something would be missing. So even I had done this thing. I have gone through extensive training in bioanalysis. Only reason was I wanted to understand ADME in a better way. So that's what I call it as that bioanalysts are forensic scientists. You know, why do we say forensic? Because after the event has happened, a murder or something has happened, then the forensic scientist job starts. I'll explain you in a while here, because an event has happened. While the event is happening, you still have a chance to modification. And once you do that, you can still wait and see the outcome. Okay. In other words, let's take an example. You can make a formulation. You can prepare a formulation and dose. Suppose after one animal dosing, you realize that formulation has some untoward effect. You would like to modify your formulation. Then you dose. Then you wait for the final study to be over. But now bioanalysis job starts here when you already give them the sample. So the whole event is over. Now they are trying to dig what exactly is the concentration of the drug or the metabolites or whatever analyte of your interest is there. So that's why we call it as forensic science. But it plays a very important role. Friends, every single assay or many assays which I may not have talked about, from early drug discovery all the way to safety studies, every single assay, I think bioanalysis has a role to play there. Okay, because all this thing, the analysis part is always bioanalysis. Okay, so the whole mission for the bioanalysis always should be to generate a very accurate and the precise data. You can have an in vivo sample, you can have an in vitro sample. There are so many challenges, of course, as you may understand, because bioanalytical methods, if you are in a discovery setup, many times selectivity is an issue, accuracy is not that great, sensitivity is not that great, so you have to work through that. Okay, there are vast diversity of analytes. You give me a plasma sample, there'll be 100 analytes to start with. So to get rid of all those, to concentrate on the one which is of our importance, that itself is a huge task. Sample cleanup, whether you do just simple protein denaturation, you do liquid extraction, you do solid phase extraction, but that's a huge task. Okay, so that's why I say there are so many challenges. But nowadays, if you want to align yourself to industrial research in current scenario, this is what, guys, you need to do. every assay, whatever I talked about, they're all now platable. Okay, they're happening in one or the other plate, and finally those plates get into mass spec or an HPLC for analysis. Okay, so this is one step towards high throughput screening. Earlier it was not possible, but it is just high throughput. Many times students feel amazed because when they're doing their studies, their entire analysis of PK analysis of 3-4 months for a molecule just by 2 routes is happening in industry setup in overnight analysis. You got my point? I'm not undermining your effort, but I'm just saying that this is how high throughput fashion it happens in industry, right? So we need to align ourselves to that. We need to understand those concepts. So it will be easy for you when you get into industry, okay? So a sample can be form of any of the things, all right? So all these matrices are available. I won't spend much time on that, but high throughput screening, this is how it happens. The moment you dose, you have a sample list. When you dose, you have a 96 well-placed format, you vortex on a high throughput like TomTech or any other liquid handling machine. You process the samples, you put it into autosampler, gets into mass spec, get the data, put it into Excel, calculate using Winonline, and you have the data ready. So this is the whole cycle. Although I took a hell lot of time to explain you all these things, but this is how it happens, right? It is so pretty straightforward process. So in summary of ADMET in drug discovery, friends, this is what I want you to understand. New chemical entities, whatever comes from medicinal chemistry, physical properties is what you need to align, what you need to screen first for. You should work very closely with the biologist. Look for the potency. It should be highly selective or whatever it is, specificity. Finally, as a DMPK scientist, work on characterizing the properties, getting rid of all the liabilities early in drug discovery. Do whatever in vitro in vivo experiments are there. Whatever sample comes out of your experiment, you should be able to interpret that data, report it back, go back to the medicinal chemistry guy. So suppose 10 molecules or a scaffold, you're given a data on 5 molecules. The chemist should be able to now modify that and come back to you with another set. So this is how the whole cycle goes on and on for different projects in the industry. Okay. Sometimes, and many a times, this what happens. You don't have to wait for this whole cycle. You can go back from here itself, and then so it's an interlinked process. I won't say DMPK, but DMPK, a biological scientist, a toxicologist, a chemist, a bioanalyst, everyone needs to work hand in hand very closely. Only then the novel drug can come very early into the market. Okay. Since my affiliation in last 2 slides, since my affiliation is with Life Science Sector Skill Development Council, I was appointed as state coordinator a couple of years ago, and last year I was fortunate and honored they have appointed me as a cluster head for the entire Western and the Central region under Life Science Sector Skill Development Council. So, Honorable Prime Minister on his inaugural lecture speech on Red Fort, he mentioned that skilling is building a better India. If we had to move India towards development, then skill development should be our mission. So, so since then, I've seen a lot of changes happening. So very quickly, maybe some other time I will reach out to all of you guys, but just to explain you, under Life Science Council, actually entire life science comes. So it could be pharma, biopharma, biotechnology, clinical CROs, preclinical CRO, API manufacturing, every aspect of life sciences comes under council. So what is the job of the council? Basically is to make the industry meet academia, all right? Because I think gone are the days when we can work in silos. In the academia need to work closely with industry. Industry needs to work closely with academia. I hear this very often that we compare Oxford University or MIT or a Cambridge University, they're doing great. But in our country, of course, this is earlier scenario. Now I think things have changed and I'm seeing many institutes doing really great. But actually there is a gap between academia and industry. We are working in different directions. And that's why we don't see this sync coming together. If we work as per industry and industry work very closely with academia, then I think the skill development will be even more enhanced. So the job of council is basically to come up with job roles. We have mapped. We have developed national occupational standards. So we have come up with around 100 job roles which are across pharma industry. These are required, and then we are training the trainers across. So I would request all the institutes, industries to participate in this, because we certify them from the council, and the aim is to have number of skilled workforce in place to the range of 2.15 million as early as possible, because we want to make this country a skill India. Okay, so qualification packs is nothing but it includes the entire course curriculum for each of the jobs. Just to very quickly, if I want to explain that, if you feel that you want to develop a job role for a animal attendant, okay, it's a basic qualification, maybe 12th pass or a graduation or a 12th, suppose consider 12th fail. So for that job role, what are the key soft skills as well as shop floor skills are required? So that forms a qualification pack. It gets validated from more than 30 industries, and then we actually make it a qualification pack, and it will be uploaded on the portal, and then onwards it becomes a national occupation standards and a qualification pack for the role of what do you call the animal attendant. So the advantage is now it is a countrywide same job role. So whenever any other country, if they want to look out for the qualification pack for a lab attendant, if they go to Life Science Council and they look for this, they find this standard on that. So that's the advantage of this. So any job roles can be developed. Especially we encourage education universities and institutes that you can develop. We can help you do that, because by now, let me just tell you that Life Science Council is biggest life science council across the world, which is operational in India. So I would wish that more and more pharmacy and veterinary colleges also become part of it, and then we can together enhance more and more skill development. You name the industries, most of the industries are part of it. Of course, research companies are less, but we are working towards that to bring them on board. And students can get a great benefit actually through this single forum. We try to provide them training. We try to align them to industries. Once they enroll we have introduced a concept of apprenticeship into pharma, which was not there, it was there, but which was never practiced earlier. So we from the government perspective, there are a lot of advantages, a lot of financial help given to the institutes and the industries who takes apprentices. So we encourage that. So students get benefit for industrial training. So, and very soon we are working very closely with CDSCO. So they are going to come up with an advisory or a notification where they would want that every workforce which is employed into pharma, be it research, be it CROs, be it manufacturing, they should be LSSDC certified, so that we can have a common system across country. So that's we are doing as a part of Skill India mission. And these are just examples of few of the job roles which I already mentioned there, and many more needs to be developed for that. With that, thanks a lot for giving me this opportunity, and thank you for the patient hearing. And I would just end here, and I would be more than happy to help you with any questions if you may have. Thank you everyone.
Thank you so much, Dr. Deepak Barot. It's an extensive presentation, and the information that you have presented shows how experienced you are in this area. And I think your material is going to be a kind of a reference material for us, especially for the students who have attended this lecture. And we will also, as a teacher, we will ask the students to refer your lecture again and again to understand various aspects of ADME. And I think your material which you have is almost, can be catered to the need for a one-day workshop, I believe. So much is there in that presentation. Anyways, thank you so much for that. And we have lots of questions in our chat box. So what I would do is I would ask only a few questions considering the time.
Deepak, thank you very much. Deepak, thank you very much. I am so happy. I have gone through your whole presentation. And very much useful to all the participants, particularly the youngsters. Okay, thank you.
Yeah. So,
Dr. Srinivas would suggest that, in the interest of time, but please share all these questions with me, and if possible, I would like to respond back to all of them in writing, and then you can share the answers with all of them. So
Yeah, sure,
go ahead.
sure. Yeah, I have already copied all the questions in a notepad, so I will be sending you the file. So you may answer it, and then we can put it in the common our ISVPT website, so that those who have asked questions can go back and ask. See the answers. Okay, so let me go through the few questions quickly. First question is about the plasma stability. If you store the plasma immediately after collecting from the animals, like PK studies, at minus 20 degrees centigrade, still do we need to do the plasma stability assay, as the enzymes may not be active at that temperature?
Absolutely. I think it's a good question. So I would say just go by the nature of your drug. You know what? I would just say that I've worked on quite a few many pro-drug projects. And what happens is, from the time you collect your sample to the time when you keep it in minus 20, that duration of a minute or less than a minute is sufficient enough to bring about hydrolysis. Okay, sometimes, not always. So I would say it will vary from case to case basis. But since you have asked, we used to work on a pro-drug program, and this is exactly what we used to do, because we knew that esterases are actually going to degrade the drug. So immediately we used to put it into ice, because minus 20 is still not possible immediately, because one person is doing or a couple of guys are doing dosing and collection. You would like to collect it on ice, and then immediately you can transfer to minus 20. But having said that, you should always do this experiment prior to doing an in vivo PK study. You got my point? I would say don't even wait till you dose the study and then do this. If you feel your drug is stable enough, and within 1 minute nothing is going to happen, please prove it just by doing a plasma stability assay. And let me put it this way: when you develop a method, basically for quantification, a bioanalytical method I'm talking about, what you're going to do, you will actually be trying to quantify your drug in a plasma, right? The matrix is plasma. So I would suggest, while you're doing the method development, you can just take a couple of concentrations additional, and when you run your initial calibration curve, you just put this few concentrations there at different time intervals. You will have your answer. So I would say don't just do it in retrospection, but do it prior. You got the point, because I get your point. But once you do the study and then you try to establish that it did not, whether it degrade or not, it is better while doing method development, because your again matrix is same. You're anyway going to add your drug. Just add in replicates, couple of concentrations, different time points, your plasma stability is done. So that crude experiment will actually help you to understand whether your drug is stable or not. And your learning from that, by all means, you apply in your in vivo study. Or you want to put it immediately into minus 20, or you may not have to do it that way. You know your drug is stable. Why do you want to do it at minus 20 immediately? Do it after 10 minutes, 15 minutes, whenever your whole blood collection is over.
Okay, yeah, that's great. Next question is Dr. Vinay Kant has asked a lot of questions. I will select only a few questions from that. I'll be sending all the questions to you. So in that, in the in vitro drug release, what should be the ideal pH of the medium, outer and inner chamber, and which solvent should be used to prepare the standard curve of drug in this assay?
Okay, so again, so this is, I will share a brief protocol, so that will answer your questions. But generally, whenever we used to maintain physiological pH, okay, because that's what is going to mimic the real conditions. So we prefer to have a physiological pH to be maintained, and even temperature also, wherever possible, it has to be 37 degrees. Phosphate buffers we have used, but again, it has its own challenges. Because if your drug is having solubility issues, then what happens is it is going to be completely crashed the moment it sees the phosphate buffer. But still, it is used extensively. Of course, it will be a little challenge for your bioanalytical scientists, because whenever they use buffer, bioanalytical scientists, what happens is they use mass spec. So because of this buffer, there will be a lot of inhibition for ionization. so their response they get would be very low. But having said that, still I would suggest we stick to phosphate buffers, and better that a bioanalytical scientist can work a little bit in enhancing his performance of the instrument in spite of using buffers. And there are ways to do it actually. They can use ionizing agent in their buffers, in their mobile phase buffers, and they can avoid this. But we use phosphate buffers generally, and I'm sure you're talking about cell-based assays. So buffer is always phosphate buffer most of the time.
Okay.
But I will share a few other options also through the protocol, which I'll, so it will be helpful.
Okay, sure. Yeah. And the next question from him only, how to conduct PK studies in rat? He's asking about, is it possible to collect large number of samples from the same rat for PK?
Absolutely. I mean, it is definitely possible. I would say there was a time almost a decade ago when I used to work in lab very routinely. We actually, came up with a concept. Not our concept; it was already a practiced concept, but we tried to do that in our lab. It was called as cassette dosing. Okay, so you're talking about single rat, single molecule. I'm saying we did multiple molecules in the same rat. It is called cassette dosing. Of course, analytically, it is very challenging because you have got so many analytes there, but it can be done exactly. And then there are advantages, pros and cons for this. One of the important advantages that you get to see the performance of your drug, the screening of 5 molecules at a time. The con side is if you don't have idea about the DDI potential, you may underpredict or overpredict the PK properties of your compound of your interest. So that's why it was not the cassette dose. Nowadays, nobody is doing it. But initially, just to reduce the number of animals to be used, cassette dosing was done. Now, to answer your question, yes, you can very well do it. I would suggest the major challenge here, because earlier we never used to think, but now with more and more understanding of animal welfare practices overall, scientists have understood. So see, why your question might have come from the blood volume, okay?
Yeah. Yes.
How much blood volume we can withdraw. So the point here is now analytically that many techniques are available, like dried blood spot analysis. You can do microsampling. So all you need is hardly 10 to 20 microliter of your sample for analysis. So what happens is when you need so less a sample for analysis, you can actually collect very little. So when you consider a rat generally we say that we should not collect more than 10% of the volume of the total blood volume, right? So you can very well dose a rat and collect less than 1 ml. I mean far less than 1 ml blood and hardly 10, 20 or 30 microliters for each time point you can select for analysis. So your entire study can be done in just 0.5 ml of plasma. So it is very much possible to do it from a single rat. Having said that, if you ask me for a mice, answer would be no. That yes, because from mice, again, smaller species, smaller volume. So again, 10, 20 microliters can very well be used, but you'll have to use more than 1 mice for sure for different time points. Okay. But if you would have asked me this question earlier, from a welfare point of view, I think we were not there as a scientific community. We were really very, very rude to these animals by collecting a lot of blood, and scientifically we were incorrect because you collect more than 30–40% of blood volume. I'll never believe your PK data because now it's not normal PK parameters you're looking for. So yes, we should stick to less than 10% of blood volume and use the recent microsampling DBS techniques. You can collect very less samples and still you can generate beautiful data out of it.
Okay, fine. Thanks. And another question by him is like, is there a requirement to take permission from CPCSA for in vivo PK studies in large animals, example, calf and goats, or permission from IAAC is sufficient?
No, absolutely. You need to. I mean, to best of my understanding, you need to take IAAC can refer. You can of course apply to IAAC, but IAAC will refer to large animal committee and they need to approve. So IAAC can only, Institutional Animal Ethics Committee can only approve small molecule protocols, PK protocols, which include all rats, mice, hamster, rabbits, everything. But the moment you get into calf and all, it has to be referred to large molecule, large animal committee.
Okay, fine. And a question from Dr. Kasturi Devi. She has asked, the microsomes collected from slaughterhouse, that is sheep, poultry, pig, liver samples, can be used for in vitro metabolism studies?
Interesting. Good question, doctor. I mean, I appreciate this because even I thought of doing this at some point in time because I thought it would be useful. But you can definitely do it. But please understand, there are strict protocols if you want to come up with a good lot of microsomes. Okay, so in a slaughterhouse, if it is under controlled conditions, okay, if you have access to that liver in a shortest possible time, so that it is immediately kept in a liquid nitrogen, or you can immediately freeze it. Those things, if you put into play, then I would suggest yes. Of course, there are few challenges because perfusion will not be great because to allow a perfusion in a slaughter animal may not be allowed. But if it is allowed, that is fantastic. Otherwise, liver perfusion, exogenously we can do it after collection. It will not be a accurate perfusion, but you can always do it and prepare microsomes. And I think this is my suggestion actually to all the students that please do this. I mean, it may not be accurate because it has to be standardized, because from an industry perspective, it has to be reliable, reproducible every single time I do it. So yes, you can do it, but I agree with you. It has to be introduced and you should do it to understand it because there'll be some challenges with optimization of the protein levels. But I think after a few cycles, once you arrive at a reasonably reproducible environment with the large animal microsomes, you should be able to do those in vitro assays. I think for the understanding for the students, it would be a great assay. And we can, of course, we can connect offline, and I will share few thought process on that exactly how going forward that can really become a research, that can be taken as a research problem for a college or a department.
Okay. Yeah, thank you so much. And for determination of pharmacokinetic parameters, which compartment model is the best? How the calculated parameters can be correlated with cell signaling leading to therapeutic response? I mean, Dr. Kaushik Kumar. Yeah.
So very quickly, and then I'll give a detailed response when I respond in writing. I would just say that from an industry perspective, if you're asking me, from a screening tier, it is always non-compartmental. Okay. The reason being, whatever platforms we are using, the softwares in terms of non-compartmental models or V-non-lane, of course, there is a compartmental modeling also available. But please understand, you are actually working with different molecules, okay, from different scaffolds, and we are subjecting them to similar PK profile. But molecules can vary slightly in life based on their solubility, based on their permeability characteristics. So if you try to do compartmental modeling for each of those molecules, actually it amounts to a lot of time time requirement. So to ease that thing, ideally what we do is we consider every study in industry as non-compartmental, okay? And then we calculate so that we have clear comparison of all the parameters, apple to apple. Because when I have a drug which I try to do it for compartment versus one drug which I do it for single compartment or dual compartment, then the comparison is not apple to apple. So if I have to screen the molecule and select a better molecule, it may not be a correct way of doing it. So the ideal way for comparison for screening is go ahead with non-compartmental modeling. Okay. Having said that, there is no harm in selecting compartments as well. If you have a well-defined structure, well-defined graph coming out of your plasma versus time concentration, where you have at least 3 points for absorption, at least 3 points for distribution, and similar points for elimination, you can select beautiful compartments out of it. Unfortunately, because in a screening assay in industry setup, we are doing a quick study with fewer time points. Sometimes we don't have liberty to choose that many time points in each of the phase of absorption, distribution, and elimination. So we don't have that confidence to select a compartment. So that's the answer that please, when you're doing a screening, go by non-compartmental. When you're working only on one molecule and you have sufficient liberty to choose the entire phase of absorption all the way to elimination, metabolism, distribution, please go by compartmental modeling. And I'll give you a detailed response for your, you said, what was the second part of your question, that for signaling pathways, which is the better molecule? Okay, so I will share a few details on that as well. Thank you.
Okay. Yeah. So I think many questions are there. Few questions. I'll just finish this. And one question. There are few participants have asked about the herbal pharmacokinetic aspects of it. So one participant asked, what are the series of in vitro assays to be done for predicting pharmacokinetics of plant extract? The other person has asked how the in vivo, herbal pharmacokinetics can be done. So a kind of an overview of herbal pharmacokinetics, if you can give, to be useful.
All right. So once again, great question. I wish I was having a simple answer to it, but unfortunately, there is no simple answer to it, because herbals, I have worked on several herbal projects in last 7-8 years. And as you know, herbal to start with, you have many molecules to work with, right? When I'm talking about so many assays, our target molecule is only one, and active ingredient is only single, at the most two. So it was pretty straightforward, because the quantification becomes easy, analysis becomes easy. When I say herbal, you already have to start with at least 20 analytes, right? So that's a challenge. Analytically, it's a huge challenge, because you can do in vitro assay. Every assay which I mentioned can be done for your herbal drugs as well. But we have to focus around, I would suggest that please refrain from using any of the in vitro or in vivo assays for herbal till you zero down on your active, pharmaceutically active ingredient. Okay, so that active ingredient, if you have zeroed down, it could be one, it could be a mixture of 2-3 constituents. Once you have more understanding on that, that is the time, go for any of these assays. Do microsomal stability, do hepatocyte assays, do induction assays, as I have shown in one of the slides. You can do CYP induction, inhibition, potential of any of the herbal extracts. But now herbal extracts, just I'm sorry, I'm taking a little extra time, but it's important to explain. Whenever we say extract, it's an aqueous or an organic extract. The moment it's an organic extract, you're going to use ethanol, you're going to use methanol. All these organic solvents, they are inhibitor themselves. So that becomes a slightly challenge. So when you do this, just make sure that get rid of all those residual organic solvent residues from your extract, and then do any of these assays like CYP inhibition, induction. All these assays can be done as an in vitro, and an animal study, you can do it. But as I said, until unless you don't, you have not characterized completely your molecule, single active constituent or a group of active constituents, please do not get into animals, because you will, if you do not derive anything from that, then it is a wastage of life of that animal. So I would suggest for herbals, because there are some challenges from analytical perspective, wait till you characterize your formulation. Once you have characterized your formulation, by all means, you should do in vitro assays like hepatocyte studies. You should do CYP inhibition, CYP induction, even permeability, you can do it. And finally, animal studies, by all means, you can do it.
Okay, fine. Thanks.
I hope I have touched base the aspects. Thank you.
Yeah. So last couple of questions. One is about the my favorite question also, like what about the zebrafish to be used as a model for PK studies? And another question is, the last question is about the in silico model, and what kind of lead do we achieve through in silico studies in PK? predictions. So
Fantastic.
first is about zebrafish.
First of all, my sincere apologies actually for not including zebrafish, and I knew Dr. Srinivas would ask me this for sure. But
My question, but somebody else has asked. Anyway.
Yeah, so let me answer this. So yes, zebrafish is a beautiful model. I guess for a very, very long time we have ignored this model, and that's why you will see in my presentation it's not a part of it. But having said that, I think it's a miss on my part, and it should be in a model. Zebrafish, I think for students, the first model to learn at your level, please do this. Okay, and I would go on record to say this because I have my initial training and exposure to zebra model at Tanuvas, where Dr. Srinivas is there. They got a beautiful facility for this. And the advantage for zebrafish is all you need is just one water tank and you are done. That's it. And you can actually have this model right in your lab. Your students can practice that. There are so many advantages. The entire life cycle of the zebrafish, as you all know, is like within our control. And being a transparent nature, you can actually see the organs from outside. And then there are so many advantages. Of course, there are few disadvantages, but as a preliminary model to understand few ideas on ADME and few on toxicity, I think zebrafish is the best model. So I would suggest all the pharmacy colleges and the veterinary colleges representatives who are there in today's call that please, if possible, develop this model, because this would be a first exposure for your students towards principles of DMPK and some safety aspects. And I think zebrafish is a real good model. So yes, it can be used in a screening tier now. And of late, I have seen, of course, if you ask me a few years ago, we never used to go for a zebrafish model. But in last 3-4 years, I have been a part of few programs where I've seen that zebrafish is definitely used as an initial model. And I guess you can correct me, Dr. Srinivas, you don't need a permission either for zebrafish model, if I'm not wrong.
Now they have made it like mandatory to go for permission. Recently, it comes under vertebrate, so it's now required to get IAC permission to conduct a study in zebrafish. But it's very easy. Okay. Nobody is going to deny that being an alternative model. Yeah,
absolutely. Yeah, sure. So yes, you can be used. And then Dr. Srinivas, what was your second question?
About this in silico, what kind of lead you get in DMPK predictions with in silico models?
All right, so
that's briefly we can say.
Yes, no, very briefly. I know I've taken a lot of time, but yes, I think in silico nowadays is must. All right, I would rather say that as a DMPK scientist, before even molecule comes to you, a lot of in silico screening would have already happened. Okay, so that in silico screening at first phase, they do docking studies. They just see how best your scaffold, your protein of interest is actually docking into the structure, whether it is a reversible or irreversible binding. All those things they are studied very early at a chemistry level itself by a chemist. So there are so many softwares available. So after that it comes to you. Okay, so he has already done his part in terms of out of thousands or hundreds of molecules, coming up with only 10 scaffolds or 10 different structures which will actually work in an efficacious manner, or where they can get a high efficacy or a potent molecule. So even nowadays for the toxicity also they can look for those alerts very early. So after that it comes to you as a DMPK scientist. So with DMPK scientists, again I will share few names like which are those softwares where you can use prediction. As I just mentioned briefly, that you can do prediction for permeability for your molecule structure. If you keep, there's a lot of database available in that. It can tell you whether there are going to be any permeability issues or not, whether solubility issues are going to be there or not. And it can even identify structural alerts, because now for structural liabilities, like the structures or the substructures on your molecule, there is enough data in domain which can tell you that this has CYP inhibition liability, this has SIP induction liabilities, this has QT prolongation liabilities. So those structural alerts can give you prediction very early. Okay, so of course it is useful in silico, but where the challenge you have is the prediction of actual DMPK properties. Okay, if I have to predict from a clearance from a rat or a mouse to the human, for the predictability of this in silico model is still somewhere around 0.5 or less than that. But still, I would say still it is better. At least it is better than having nothing. So that predictability of accurate parameters across species of this in silico model is still questionable, but no denying the usage of in silico models. It can definitely be useful. So eventually you will have to do in vitro and then at some point in time in vivo to have the best advantage. And in less than 10 seconds, let me say this. Many students do ask this question that is it that because of the predictability and our closeness, like if we do it in monkeys, the data would be very close to human, or if you do it in dogs, the data would be very close to human. Let me tell you, friends, unfortunately, from our experience, it's not like that. Okay, so otherwise, every study would have been referred for a monkey study, but it doesn't happen because the SIP isoform expressions are different in different species. Sometimes you may have good correlation from mouse to human. For few drugs, you may have good correlation from rabbit to human, or sometimes you may have from monkeys to human. So that's where it's very difficult to predict just based on the allometric scaling of surface area. Okay, so I hope I answered your question.
Yeah, thank you so much, Dr. Deepak. Now, may I request Professor Sailesh Bhavsar to deliver the vote of thanks.
Thank you, Dr. Srinivasan. So at the end, I would not take much time. I'm thankful to Dr. Deepak Barot, to whom I have taught, and today I have learned many things from him. So most of the youngsters, they are at advantage, and they can consider Deepak as a role model. They can develop themselves into an entrepreneur. So once again, I congratulate Dr. Deepak, as well as thank you on behalf of the ISVPT for delivering a very useful, informative webinar. I'm thankful to all the participants, especially the senior executive committee members, all the senior pharmacologists, toxicologists, including the Dr. K.S. Rao, and all the other those who are present and who have remained present. So today we have reached to the peak of 250, and so it's a very good webinar. And I'm thankful to each and every participant for their active role during this webinar. At last, I'm thankful to the webinar team, including Dr. M. Thakur. So we have worked together and made it possible to have such type of the good webinar. So I'm also thankful to Dr. Srinivasan, Dr. Ratnadeep, as well as our guru, Dr. Thakur sir, for providing us the facility. So at the end, I am thankful to each and everyone, those who are involved in making this webinar a success. Thank you, everyone.
Thank you, sir. Thank you so much, sir, and Dr. Deepak. And hello. Yeah, Dr. Deepak, you're speaking.
Yeah, so very quickly, would like to thank the organizers for giving me this opportunity, and I'm indebted to my mentor, my guide, Dr. Bhavsar sir and Dr. Thakur sir, and mentors from industry, Dr. K.S. Rao sir, and then Dr. Srini, my colleague, and then Dr. Sadaria, and all the organizing committee members of the ISVPT. For giving me this opportunity, and thank you to all the participants and for the wonderful questions you have asked. And I will coordinate with Dr. Srini and take the remaining questions, and I'll try to answer those questions to best of my understanding. But I'll be sharing. I think I've shared my email ID and all. Any point in time to all the students who are doing your masters, PhDs, or any of those people, if you have any questions, queries, please feel free to reach out to me. I'll try my level best to answer your questions. And if I don't know, then I'll go back, try to find answers for you and then come back to you. So with that, thanks a lot for your patient hearing, and my sincere apologies for a very long presentation. So thank you all.
Yeah, thank you, Deepak. It's really very informative session. And thanks for supporting your support for our students also. Like maybe you will get a lot of requests. And I'm going to send you the remaining questions to your email ID. Maybe when you're releaser, you can answer them, and we'll put it in the common forum so that everybody can access. Thank you so much. Thank you all for being here. And I'm closing the session. I may request so please allow me to end the session.
Yeah, Dr. Srinivasan, you are permitted to close the session. Right. It was very, very informative and exhaustive speech by Dr. Barot. Thank you.
Thank you, sir. Thank you.